Ski-bit-I vibes are strong with this research on carboxymethyl Chitosan and starch nanoparticles.
Yeah, this is me. You're probably wondering why I would ever trust AI-generated brain rot to explain science, let alone science that I researched. Now, if you're a friend of a channel, you know that this is something that I do. And if you're new to the channel, welcome. I'm Matty C. This is the series where I put scientific papers into PDF to brainrot.org and see if they can explain science a little bit more informally. At the end of it all, I'll score the video on its scientific accuracy, its clarity, and its overall entertainment and brain rotability. Today's paper is called Starch Nanoparticle / Carboxymethyl Chitosan Nanoassemblies for Intranasal Delivery of Antipsychotic Drugs to the Brain. Yeah, some dork made this title an absolute mouthful, but the gist is quite straightforward. We know that delivering pharmaceuticals to the brain is really tough, but can we engineer material that makes delivering these molecules less tough? Before we get too technical, we're actually going to watch the full brain rot video together right now. And if you pay close attention, you'll be able to see where it falls short and where it's shockingly accurate. So without further ado, class is in session.
Innovative antipsychotic delivery method. Ski-bit-I vibes are strong with this research on carboxymethyl Chitosan and starch nanoparticles. The team crafted a dank, nano-gel assembly that switches size in the nose, targeting the brain like a Sigma goon. Intranasal delivery of haloperidol is the edge we need, boosting bioavailability while keeping toxicity low-key. These nano-assemblies are a ship to better drug penetration, releasing 55% of the MEDS over 5 days, far outperforming the free stuff. They even showed Matty C to compatibility and suppressed rat locomotion longer than traditional methods. This is the future of brain delivery. Folks, get ready for some serious brain rot innovation.
So, what'd you think? For me, personally, I thought it had an incredibly rough start. Brain rot was trying really hard to try and explain what the particles used in this study were made of, which is carboxymethyl chitosan linked to starch nanoparticles. That is a lot of big words, but the best that they could come up with was this.
Carboxymethyl Chitosan and then starch nanoparticles.
Now, there's nothing inherently wrong with this. I understand that these types of sites and these types of models can't say every single scientific word. But two things to that. One is that if this was my first exposure to this paper, which is probably most of you, I would be immediately turned off by just how clunky all of this sounds. Now, luckily, this site does have a little script at the bottom after you've made the video, but if you're just hanging out on TikTok and this video comes up, I'm scrolling away immediately. Not to mention, there are way better models that have a better time and an easier time sounding these things out. Admittedly, I was ready to write this video off almost immediately, but on a second review, I realized the AI actually described the core concepts of the study quite well. It said that these nanoparticles were able to switch size in the nose to target drug delivery to the brain. Like a Sigma goon, of course. As the very clunky title might suggest, these nanoparticles are designed to move through the nose in order to drop off therapeutics to the brain. The size and chemistry of these particles, which are called nanoassemblies, determine how they move through the nose. Almost counterintuitively, when nanoassemblies are large and swollen, they're able to slip and slide through the nose easily. But eventually, due to environmental factors in the nose, they break apart into small mucus-binding particles that can actually stick to the inside of the nose. This not only prevents clearance from the nose too soon, but it actually enhances the slow and steady release of drugs into the brain. Again, it's that fine interplay between size and surface chemistry. And the AI got this exactly right. This is the main point of these particles, and it's actually the main point of this study. It was mostly a material design and engineering study. And amazingly, they knew that these particles were made of a mixture of carboxymethyl chitosan and starch nanoparticles, the smaller mucus-binding particles. So maybe we should give them a little bit of grace for stumbling through their pronunciation. But now you might be wondering, why do we even need to go through the nose? What was the point of making these materials in the first place? The short answer is that getting drugs to your brain is stupidly hard. Most drugs can't get to the brain because of this thing called the blood-brain barrier, which is a semi-permeable membrane that acts like a bouncer at a VIP nightclub. Don't have the very specific chemical makeup to get it to the brain? Beat it, pal. The blood-brain barrier is designed to keep foreign, harmful entities out of the brain and make sure that everything's working properly. The unintended side effect being that it also keeps out the drugs that are intended to make the brain work properly. But delivering intranasally, that's like the sneaky back entrance to the club. You can actually bypass the blood-brain barrier entirely and go directly into the brain through several different neural pathways, which are located at the back of the nose. And these neural pathways can be manipulated with specific nanoparticle sizes and chemistry, as we've briefly summarized here. Bigger particles can use one pathway, smaller ones can use another. Hydrophobic particles use a different pathway from hydrophilic ones, etc., etc. Now, if you've seen the movie Scarface, this concept probably isn't a surprise to you, but at least now you know why he did the things that he did. Now, unfortunately, this is about as good as things got for the brain rot. If you've taken other classes in Brain Rot University, you'll know that this AI on this site really struggles to provide the full context that makes people give a shit about this research. Take a look at the middle part of this whole video, for example. Phrases like bioavailability of haloperidol and calling them a ship with better drug penetration of over 55% over 5 days. At first glance, you might think, wow, the brain rot's able to pull out data. That is super impressive. But at second glance, you realize that unless you actually read the paper, none of this means anything. For example, they don't even explain what haloperidol is, and it comes right after they were hyping up carboxymethyl Chitosan and starch nanoparticles. So most people would probably think, well, what the hell is this and why do I care? Because it is the antipsychotic drug, the actual active molecule that treats different psychosis-related diseases like schizophrenia. The whole point of this research is getting this molecule to the brain more effectively. The nanoparticles are just a method of getting them there. But the AI doesn't link this to any of the other technical words that they've said up until now. And their stats are almost completely out of context. 55% over 5 days refers to this graph, where we were able to release 55% of haloperidol that was originally in the nanoassemblies. But the brain rot specifically says drug penetration. Drug penetration implies that the drug, or these drug-carrying particles, could penetrate through some biological surface, like the nasal mucosa. But that's not even what we did. We just measured how finely controlled environments in a dish could change the rate of drug release. Now don't get me wrong, there were some really cool reasons why this release happens, and much of the paper dives into how we controlled the chemistry to make this happen. But again, the brain rot doesn't link these reasons to practically anything. Now, I'm not saying that the brain rot needs to encapsulate all of these details, right? Like, it's brain rot, for God's sake, and the video is like less than a minute long. And nothing says you're a beta like showing data. And if this was just for shits and gigs, then that's fine. But I don't think you're actually helping people to learn things faster and remembering things longer, which is the tagline to your own site. These videos would be more effective learning tools if they just focused on like 1 or 2 different concepts and then expanded on those. At that point, do whatever. Skibbity the hell out of them. It's especially disappointing for this video because it also ends really strong. The brain rot is trying to summarize the most important in vivo results. The part of the study that suggests, hey, maybe one day we can use these to save human lives. Let's focus specifically on the part where the brain rot talks about suppressed rat locomotion. The known side effect of haloperidol is reduced movement or reduced locomotion, the literal opposite of what Scarface was into. We tested 3 forms of haloperidol. One by itself, this is the free stuff. One put into a nanoassembly, and another where we put it into one of these mucus-binding, smaller starch nanoparticles. We then gently injected each version into rat noses with little pipettes. And after monitoring rat locomotion for 4 hours, the data was quite clear. The haloperidol-loaded nanoassemblies could significantly reduce their movement when compared to the other 2 groups. This is exactly what the brain rot was trying to say. Suppressing rat locomotion better than these traditional or non-nanoparticulate methods. This tells us that the nanoassemblies are the optimal method, the optimal delivery vehicle to slowly and controllably release haloperidol into the brain. And we did another experiment to suggest the mechanism for this is that the larger nanoassembly has better chemistry that allows it to slip through the mucus, whereas the other 2 methods get trapped or get eliminated from the body before they can get to the brain. Again, I would never want brain rot to give like this level of detail. But when these findings are lost in a soup of unconnected scientific buzzwords, it's pretty much impossible for anyone to learn anything from this or get excited about these results. But the overall implications of this is what really matters. We already know that it's so difficult to get drugs to the brain at all, let alone finding the perfect dose between something that is therapeutic, something that helps people, and not a dose that's so high that it actually causes more harm than good. This type of material can fine-tune what dose actually gets to the brain. optimizing that balance between efficacy and harmful side effects. Not to mention the convenience of needing to take the same drug less frequently. But this got me thinking, why is this like this? Why does this AI continually do this where it can pick out certain important parts, but it doesn't know how to contextualize or put them together? Well, the obvious answer is that PDF to brainrot.org isn't going to actually read the paper. It's just choosing random things, keywords and numbers and stats that it thinks are important, throwing in edge and skibbity and goon, and then calling it a day. But when I went through this abstract line by line, I was shocked to find that the AI was actually working way harder than that. There were several times where the AI selectively excluded data that most brain rot watchers wouldn't care about. Things like this line, talking about 80% encapsulation efficiency, or some of the other really specific chemistry that was used to make these nanoassemblies. Yes, they're important to nano nerds like us, but it's very much okay that they were skipped when making brain rot for the general public. In fact, of this abstract, these would be the first two things that I would cut. Combining this with the fact that it grabbed the obviously important stuff like suppressed locomotion study and
Carboxymethyl Chitosan Nanoassemblies.
This does make me think that the AI is using some level of reasoning after it reads, or at the very least scans, the entirety of the rest of this document. Scan the whole thing, pick out its favorite parts, and then make the brain rot out of that, even if those things aren't actually linked together. That, or it's completely random and I'm just giving it way too much credit, but I'm an optimist, okay? I'd like to think it's thinking a little bit. So with all of that, with all things considered, it's time to finally rate the PDF to brain rot experience for this paper. Brain rot's ability to understand and explain the concepts at a high level were still quite good. So let's say a 7.5 out of 10. Because I really liked the beginning and the end. But the juicy bits in between, the details, nuance, context of what is happening, and more importantly, why, falls frustratingly short. I can't confidently give this above a 3. Let's say a 3 out of 10 for scientific rigor and, more importantly, clarity. And finally, I'm a little mixed on their use of brain rot. Like, sure, Sigma Goon Squad and all that absolutely kills me. I love that stuff. But I absolutely cannot stand it when they use brain rot terms like regular words, like intranasal delivery of haloperidol is the edge we need. Using edge here, but in a literal non-penis way in a brain rot video should be a jailable offense. 5 out of 10 at best for brain rot ability. Tallying up these scores, you get less than a 52%, which honestly is a little bit horrendous because I am known to be a very easy marker. Seriously. I had to have a stern talking to one time when I was a TA because I marked too easily. But honestly, for this video, I think it's fair. It was so close to being good, but then really pulled away when it had the chance. This is generally the problem with making AI-generated science content. We see this a lot at Brain Rot University where it can parrot the conclusions and some of the main concepts really well to make it sound smart, but misses all of those mechanistic details, the part of science that is the most important. And for a general audience, maybe this doesn't matter. But for this specific audience, us delectably cute nerds who want to know the nuance and know the details, it's a bit of a problem. Now, I would love to hear what this adorable audience has to say about this specific video. Did it help get some points across? Keep you engaged? Give you a little chuckle? Let me know down below. You can click somewhere on the screen here if you want to see how this video compares to other brain rot videos that explain science poorly. If you're not into AI nonsense but are really interested in learning how biomedical engineering is trying to treat the brain, you can click on this video about how a virus was used to treat Huntington's disease. You got options, baby. But as always, thank you so much for stopping by, and we'll see you later, nerds.